So in cell cycle, gap 1 synthetic, gap 2, then it enters the division phase. Now we need to discuss division phase. Division phase occurs in two phases, what is known as two types, mitosis and meiosis. So any cell, living organism that contains the cell, any cell of the living organism undergoes either mitosis or meiosis. So you have to remember two important definitions here: mitosis, meiosis. The definitions of these two you need to remember. What is mitosis? Mitosis is the type of division where equal number of or amount of chromosomes will be equally divided into the daughter cells. From mother cell, the DNA will be equally distributed. That is called mitosis. So the newly formed cells contains equal number of chromosomes in the mother cell. That's why the chromosomes or DNA will be doubled in the synthetic. Suppose 46 chromosomes are there in the mother cell. It will be divided, it will be replicated into what is known as 92 chromosomes. Then they are equally distributed into 46 in the daughter cells. So to maintain the constant number of chromosomes in the organisms, this DNA replication synthesis takes place in the synthetic phase. That process is called mitosis. In mitotic division, what happens? It maintains the constant number of chromosomes in the daughter cells. Whereas this mitosis occurs in all somatic cells or body cells. Somatic cells. In all living organisms, the cells again are two types: somatic cells and reproductive cells. In all somatic cells, this mitosis division will be takes place. Then meiosis. Meiosis is reducing division. Number of chromosomes will be reduced. It is occurs in the reproductive cells only. Here won't confuse. For that I will explain one thing. In case of diploid organisms, 2n, I will take human beings as example. 46 chromosomes are there. Both mother and father contains 46. The newly born baby should contains also 46 chromosomes. How? So in mother reproductive cells, the gamete ovum will be contains 23 chromosomes. So the ovum cell formed into 23 chromosomes through the meiosis, reduced division. From male, the gamete contains 23 chromosomes. These two are merged by fertilization and form the 46 zygotic cell. So to maintain the constant number of chromosomes in the daughter organism, the reproductive cells undergoes meiotic division. That is called meiosis or meiotic reduced division, number of chromosomes. So these two are the two types of cell divisions: mitosis, meiosis. In GATE syllabus, there is no specific on the mitosis or meiosis, but I will explain the various stages in the mitosis. You see here, mitosis, the division of the nucleus, also called as karyokinesis. It occurs only in the eukaryotes. You remember, mitosis occurs only in the eukaryotes, not in prokaryotes. It contains 4 stages: prophase, metaphase, anaphase, telophase. Mitosis contains these 4 important phases or stages: prophase, metaphase, anaphase, telophase. So the cell, the somatic cell, undergoes 4 different phases before going to divide into 2 cells. You can easily understand by seeing the diagrams. prophase, what happens? First the cell enter the prophase stage where the condensing of chromosomes takes place. This type of chromosomes condensed. Then metaphase. In metaphase what happens? All chromosomes comes and join in the center area of the cell. Mitotic. In mitotic division, the center phase. Here these chromosomes comes and in an orderly manner these are arranged at the center part of the cell, and these are attached with the spindle fibers that starts in the next phase called anaphase. Through these spindle fibers, the chromosomes will be moved toward their polar regions. These are like this. During the movement of these chromosomes, the centromere attached with these spindle fibers. Then telophase. In telophase, what happens? The chromosomes are clearly separated and division of the cytokines will be takes place. Then two cells will be formed. Two cells completely will be formed. Then before the formation of new cell, the cytokines will also be takes place. The separation of the cytoplasm. Then meiosis. I already told you that meiosis occurs in the reproductive cells only during gamete formation. Gametes formation. For example, ovum cells and sperm cells. Male gametes, female gametes. So in that cells, meiosis will take place. What is the main importance of the meiosis? To maintain the constant number of the chromosomes in the diploid organisms where two sets are there. From mother and from father, the formed gamete should contain half of the chromosomes. Suppose 2n mother, 2n male, 2n female. The gamete from male 23, gamete from female 23. These two are merged and form the 46 chromosomes containing cell. That cell develops into the organism. In case of humans, in case of plants. So in any diploid organisms where reproductive cells are there, then where reproduction is there, in that area the type of division is meiosis. So meiosis occurs only in the reproductive cells only, not in the all other parts of the organism. So here you need to remember somatic cells are reproductive cells. Somatic cells, body cells, generally follows the mitosis. Reproductive cells follows the meiosis. So meiosis contains various stages. Again here you can see this. It is the 2n number of organism. Meiosis 1, where suppose here 4 chromosome sets are there. 4 chromosomes, here 2, here 2. In meiosis 1, so in meiosis 2 substages are there, meiosis 1, meiosis 2. Meiosis 1, half of the chromosomes will be divided. Then this half chromosomes or half number of chromosomes containing cell undergoes the meiosis 2. It is equal to the meiotic region, mitotic region. It forms two cells, 2 chromosomes. Again this cell divides and forms 2 cells. So at the end of the meiosis, 4 gametes will be formed. Each gamete contains only half of the chromosomes of the mother cell. What is the mother cell gametes? Mother cell chromosome number 4. Daughter cells contains 2, 2, 2. In case it is 46, all daughter cells contains 23, 23, 23 gamete. Number of chromosomes containing gametes. So, meiosis importance is it has to be reduced the number of chromosomes in the formed gamete cells. Why? Because these cells merged or fertilized with the another gamete which contains half of the chromosomes. Suppose here 23, this 23, and form the 46 chromosome cell. So our understanding, I explained by taking the example of human beings, 46 chromosomes will be formed. So in meiosis 1 is a very, very important as per examination. How the mother cell will divide it into 2 cells where the number of chromosomes will be decreased halfly. How it will be decreased into half of the chromosomes. You see. Again it is divided into prophase 1. In prophase 1, the chromosomes are paired. Here, actually, prophase phase 1 contains very important subtopic, pachytene. In pachytene, synapses, pairing of homologous chromosomes will be takes place, which is in that crossing over exchange of genetic material between the non-sister chromatids of the homologous pair. Here I need to explain 2 important words. What is homologous pair? Homologous pair of chromosome. These 2 chromosomes are called as homologous. You see here, it is homologous chromosome. Homologous chromosomes. In each chromosome, the arms are called chromatids. 4 chromatids are present. These 2 are homologous chromosomes. This structure is called tetrad stage. 4, 1, 2, 3, 4. Tetrad stage. These 2 are homologous chromosomes and these 2 arms are called non-sister chromatids. These 2 are sister chromatids of this chromosome. These 2 are sister chromatids of this chromosome. When this chromatid attaches with this chromatid, these 2 are called non-sister chromatids. Between these 2 non-sister chromatids, exchange of genetic material will be takes place. From this chromosome part to here, this part here, it is called crossing over. Exchange of genetic material between non-sister chromatids of the homologous pair. So crossing over occurs during which phase of cell division? In meiosis, prophase 1, pachytene substage. We have to remember. In meiosis only crossing over occurs. What is crossing over? Exchange of genetic material between the non-sister chromatids of the homologous chromosomes in the mother cell. Then once this happens, it separates the 2 chromosomes. You see here, tetrad stage, this type of exchange will be takes place. So in meiosis 1 prophase phase, this substage is very, very important because of this exchange of genetic material between the non-sister chromatids of the homologous pair, variations will be takes place. Suppose the children of the a parents will not 100% resemble to the parents. Some new characteristics, variations will be takes place. This is because of the combination of or exchange of this genetic material. So in meiosis, crossing over exchange of genetic material is important word you need to remember. Then metaphase. What happens in metaphase? Chromosomes will be attached at the center point through the spindle fibers and move towards next stage, anaphase. Anaphase 1 separates, you see, two chromosomes. So at the end of the meiosis first stage with two chromosomes. At the beginning, how many chromosomes? 4 chromosomes. You see 4. 1, 2, 3, 4 chromosomes. At the end of the meiosis 1, the two cells contains two chromosomes. Next, meiosis 2. You see, in meiosis again these two cells undergoes meiosis 2. In this stage, generally, two cells are formed, two chromosomes. Totally 4 gametes will be formed. I will show this. You see, total 4. 1, 2, 3, 4 gametes are formed. So at the end of the meiosis, 4 gametes will be formed. You see the chromosome structure. This is the chromosome. This is centromere. Centrioles are involved in the spindle fibers formation. Okay. So in cell cycle, you need to remember interphase, G1 synthetic and gap2, and cell division. Okay. Later I will explain what is known as how the cell growth will be controlled, how the cell cycle will be controlled step by step, from gap 1 to synthetic phase to gap 2 and division phase. How the cell will be controlled, that is called cell control. If cell will not be controlled its division or its phases, the abnormalities will be developed in the cell. So it won't be happen because the individual cell has the capacity to control its phases and maintain the cell sustainability.